Good evening. My name is Nicholas Hanley, and today I'm going to be talking to you about the pathway of sound conduction. The objectives for this presentation are first, to explain the functions of the outer, middle, and inner ear. Secondly, I'd like to list the components of each region of the ear and talk a bit about each one of those. Next, we'll describe the pathway of sound from the source until it is detected by the brain. And finally, we'll explain how the cochlea actually converts a sound wave into an electrical impulse. Okay. To start off, let's talk a little bit about a sound wave. A sound wave is a longitudinal or compressional wave, and it consists of alternating regions of compressions, as you can see here, and rarefactions. Now, the compression is obviously where it is compressed, or the particles in the air or the media are compressed together or tightly packed. The rarefaction is where the particles are spread out a little bit more, and it alternates between these two regions. Um, the pitch or tone of a sound wave is determined by its frequency. Um, the higher the frequency, the higher the pitch. For human beings, our ear can detect frequencies from 20 to 20,000 hertz, with our most sensitive area being between 1,000 and 4,000 hertz. Next, the intensity or loudness of the sound is related to the amplitude. The amplitude in a longitudinal wave, the amplitude is determined by how compressed the compressions are. You think about, like, I have students that bounce pens up and down, the clicky pens, and they'll push it down. The further they compress that spring, the higher the pen is going to jump. So that's basically what we're doing with intensity or loudness. We're measuring how compact that compression is, and the more compact it is, the more energy it has, and the more intense or the louder the sound will be. Finally, the timbre or quality of the sound. It depends on how the different frequencies are superimposed over each other. And basically, this is the quality that actually gives everybody's voice uniqueness to it. So it's how you can determine or understand if it's your daughter or your mother or your grandmother talking to you. They have different tones and timbres to their voice that we can recognize and use for identification purposes. Okay, now that we know what a sound wave looks like, let's go and talk about how the sound is actually conducted. To do that, we're going to talk first about the parts of the ear. So if we look at the human ear, it's divided into three regions. We have the outer ear, the middle ear, and the inner ear. And really physically is how they're separated out. So your outer ear, its main function here is to collect the sound waves from the air and funnel them down through here to get to the middle ear. The middle ear is responsible for conducting the sound from the outer ear portion to the inner ear, and as it's doing that, it is going to have a role in amplifying the sound as well. And finally, in the inner ear is where the big bulk of the process occurs. This is where we're actually going to convert the sound from a sound wave into an electrical impulse that can be detected in the brain. Okay, we're going to look at each part separately now. If we look at the outer ears consist of three parts: the pinna, which is the part of it that you actually see on the external side. The canal that runs here, it's the ear canal, or the fancy name for it, external auditory meatus. And then the tympanic membrane, or the eardrum here, is the third piece. The tympanic membrane is kind of the separating barrier between the outer and the middle ear. And again, the goal here is to collect the sound from the environment and guide it through to the middle ear. What's unique about this is the pinna, the design of it actually kind of blocks sounds from behind you, allows sounds from in front to go in a little easier. That difference is what we use to kind of help locate where a sound is. So someone who only has one functioning ear has a little more trouble locating where the source of the sound is, whereas somebody with both ears can do that. Humans, we're not able to move our ears. If you see dogs and cats and things like that, they can actually rotate their ear to line up with where the source is, but we don't have that ability. Okay, now the middle ear. Okay, the middle ear, okay, like I said, the tympanic membrane separates the two, and it's a tight, stretched covering that separates the outer from the middle ear, and it's actually going to be used as the sound wave hits it, the air molecules that are compressed, the tympanic membrane eardrum is going to vibrate. When it does that, it causes movement in the malleus, the incus, and the stapes, the three ossicles in the middle ear. They're little tiny bones that go through and help transfer the sound from the eardrum to the middle ear, to the inner ear. This little tube you see running down here that's not fully in the picture is our eustachian tube. It's really important that the pressure in front of and behind the tympanic membrane is balanced for the vibration of sound to work properly. So your eustachian tube is what does that. We want both sides of the tympanic membrane to be at atmospheric pressure. And since the outer side is exposed to the air, atmosphere pretty easily, it's easy to do that. The inside here, the eustachian tube, is what allows us to depressurize that or maintain that pressure equilibration. So if you've ever had your ears pop after traveling on an airplane or through the mountains as you're driving, that's just this or the eustachian tube opening up. It's normally closed, but it opens up. Opens up, and that popping sound you get is that pressure equalizing. Now, we can also cause that, help induce that process by yawning, chewing, all that stuff. So that's why they recommend gum on planes or as you're driving in cars. The other role here is kind of important, is that it's going to amplify the signal between here, because the cochlea here is filled with fluid, and it's harder to make fluid move than it is air particles. So it's going to amplify that signal in two ways. Number one, the tympanic membrane is much bigger than the oval window, so surface area wise, we're going from a big surface area to a smaller one, and we're applying a force on it. So if you apply the force to a smaller area, you actually get more pressure there. So that amplifies the signal there. And then the second thing is a mechanical advantage. These bones act as levers, and that gives the mechanical advantage and increases the amplitude of the sound wave. Overall, we increase the force by about 20 times. And now the inner ear. Okay. The inner ear, the components here, the main components are our cochlea and our auditory, our cochlear nerve. We also have these semicircular canals. Those play more of a role in balance and equilibrium, which is the third bullet point here, and that's for a different conversation. What we're going to focus mainly on today is how the cochlea here, this little seashell or snail-shaped looking thing, converts the sound wave into an electrical impulse that our brain can detect. Okay, so if we unroll the cochlea, it kind of helps us. It's coiled up, but inside of it, as we see here, there's three fluid-filled compartments. We have the our the cochlear duct, which is the center part here. Sorry, my mouse wasn't working there. Our cochlear duct. We have our scala vestibuli and our scala tympani. And then inside we have two membranes, our vestibular membrane, which you can see a little better in here, and we have our basilar membrane, which is the orangish one here. So if we go to the next slide, we can see a little bit more there. We're going to zoom in here on this organ of Conti. If we look, we have this basilar membrane, which is represented in gray here, that supports this organ of Corti. The organ of Corti contains about 15,000 auditory hair cells per cochlea, so 15,000 on each side, 30,000 total. And on here, the hair cells are arranged in rows. Okay, we have an inner hair cell that's a single row of hair cells, and then we have our outer hair cells, which is three rows in a row. On top of them, we have our stereocilia, which if you can see in here, inner hair cells, and we have these here is what we're looking at. The stereocilia are mechanoreceptors, so as they move, they're going to trigger electrical impulses. The stereocilia are the actin-stiffened microvilli that protrude from the surface of each hair cell. And there are about 100 of them per stereocilia. The tectorial membrane here sits above it, and the hair cells, these stereocilia, are contacting that. Um, so as the stapes moves on the outside and contacts the oval window, there's a pressure wave that's going to move through the upper compartment, and that can dissipate some of the pressure by displacement around the round window and deflection of the basilar membrane. So as a sound wave comes in, this basilar membrane right here is going to move. And as it moves, it's going to cause these hair cells, its stereocilia, to move as well. Um, um, so that movement is going to trigger an electrical impulse because these hair cells are mechanoreceptors. So if we look a little more closely at these micro or stereocilia, we're going to see a little bit how this works. So here's how they're organized. Okay, at rest, the stereocilia are straight up and down. But notice that the inner hair cells transform mechanical movements into electrical impulses. If we look at these stereocilia, they're arranged in rows of 3, with each row slightly increasing in height. So see a smaller one, and we move up in height with the hair cells. Between these hair cells, you have little bands connecting them called tip links, and they're going to link these hair cells together. And then this tallest stereocilia is going to be connected in direct contact with the tectorial membrane above it. Now, if we stretch these tip links towards the tallest one, that's going to force some voltage gated or mechanically gated cation channels to open up. And the endolymph, the environment that these hair cells are submerged in, is unique in that it has a higher potassium ion concentration than the inside of the hair cell. So when these mechanically gated channels open, the potassium ions are going to move into the hair cells and depolarize it. If the basilar membrane is moved in the opposite direction, like we see in this third picture, the hair cells are going to tilt towards away from the tallest one, and that's actually going to cause the tip links to unstretch or slacken, and that's going to cause the mechanically gated ion channels to close. That would decrease the amount of ions, potassium ions entering the cell, and hyperpolarize the cell. Now, these hair cells don't undergo action potentials on their own. They communicate via chemical synapse with the terminals of the afferent nerve that kind of conglomerates into the auditory or cochlear nerve. Some neurotransmitter is released by these these synapses via calcium-induced exocytosis in the absence of stimulation, but the depolarization of the hair cells opens more voltage-gated calcium channels, which results in additional calcium entering and increasing the rates of the neurotransmitter secretion, which will step up the rate of firing in the auditory nerve. Conversely, if the rate of firing decreases, resting levels hair cells, they're going to release less neurotransmitters because those voltage gated or mechanical gated channels will close, and so less neurotransmitter is produced. That hyperpolarizes and causes them not to send signals. Next, we're going to talk about, basically, if we summarize before we get to cochlear implants, so we detect the sound wave from the pinna, direct it through the ear canal to the tympanic membrane. As the tympanic membrane vibrates, it causes the ossicles to mechanically move, which taps on the oval window in the cochlea, moving the fluid around in there, and then that fluid causes these hair cells to move as the basilar membrane moves and triggers the electrical impulses that are then sent from the auditory canal to the various portions in the brain. Some of the portions are the primary auditory cortex, as well as the brainstem and medial geniculate nucleus of the thalamus. So various places it can be sent to. Finally, to wrap things up, just a medical thing that kind of ties into this are cochlear implants. It's a project I actually give to my class. They have to discuss the benefits of getting a cochlear implant if they would go deaf on their own as 16-year-olds. But basically, a cochlear implant is designed to bypass the cochlea, so it directly ties into the auditory nerve instead of using all that stuff. So most people with sensory neural hearing loss are the ones who get these. Research shows that the younger you are when you get the implantation surgery, the better your results are going to be. You're going to see, basically, you're able to pick up on the language and you're going to talk more normally and all that because you're around speech more often and you develop around the same rate as other children your age. Again, the younger you get it. The older you are, the harder it is for your brain to especially if you've never heard sound before, the harder it is for your brain to distinguish specific sounds and isolate the things you need and not get overwhelmed by all the background noise you see every day. So cochlear implants, a wonderful invention, helps with hearing loss in many people, but can be controversial at some times. Okay, so to review, our objectives were to explain the functions of the outer, middle, and inner ear, to list the components of each region of the ear, and then describe the pathway of sound from the source until it's detected by the brain, and then to give a pretty good explanation about how the cochlea converts a sound wave into an electrical impulse. Here are my references, and I thank you for your time.